Hello friends, in this video we shall discuss about few questions about cell cycle and cell division. Now, cell cycle. Cell cycle is nothing but the cyclic events occurring since the birth of a cell till it divides. It means it is cyclic, it is nothing but the cyclic set of events that occur the moment a cell is formed. Okay, now suppose this is the formed cell. Okay, two cells are formed at the end of a cell cycle. Okay, now these cells, this is with respect to mitosis, so two cells will be formed from a single cell. Now this cell again, okay, it enters the next phases of the cell cycle and finally it divides into two. So here this cell is formed. Okay, now this cell is formed here and again it undergoes several changes. Okay, it undergoes several phases and finally two cells are formed from this cell. Now this is nothing but cell cycle. Now cell cycle is basically divided into two phases. One is called interphase. Okay, one is called interphase, so that extends from G0 phase up to G2 phase. So this entire, okay, sequence here you can see from G0 phase, G1 phase, yes, G2 phase. Okay, now all they come under interphase. And the second phase is M phase. Suppose if the cell cycle is 24 hours, then 23 hours, okay, cell remains for 23 hours in interphase. Okay, and only 1 hour, okay, is available for M phase. Okay, now let's see what happens. Now during interphase the cell prepares for the division. During interphase cell prepares for the division, and during M phase cell actually divides. Okay, so M phase means here mitotic phase, where cell divides into two. Okay, so interphase is the phase, okay, where the cell starts prepares to divide, and in M phase cell actually divides. Okay, right. Now first let's discuss about interphase. First part, okay, in interphase, or you can say the first phase in interphase is G naught phase. It is G naught phase. So G naught phase is the phase, okay, where cell has not decided to divide. Okay, it remains no stagnant here. Okay, it remains stagnant doesn't mean it remains stagnant. The activities of the cell will be going on, but cell is not preparing to divide. So if a cell is in G naught phase, it means cell is, okay, not preparing to divide. Okay, it is doing its regular functions, and whenever the cell decides to divide, then it enters the next phase of the interphase, that is called G1 phase. Now in G1 phase, okay, most of the cell organelles they are duplicated, and certain proteins which are required for the next phase, that is S phase, they are synthesized in G1 phase. Now here G refers to gap. Now, this is gap 1 phase, this is gap 2 phase. So in gap 1 phase what happens? Most of the cell organelles they become double. Okay, cell grows in size, and also the proteins which are required for the next phase, that is S phase, they are prepared in they are produced during G1 phase. Then comes the S phase. Now during the S phase the amount of DNA doubles, because DNA replication occurs during the S phase or synthesis phase. Okay, so DNA becomes double here by the process of replication. So if the initial value of DNA is 2C, after S phase it will be 4C. Okay. If the amount of DNA is 2C before S phase, during and at the end of S phase the DNA becomes 4C. That is the DNA becomes double. The DNA becomes double in S phase. After S phase, next phase is G2 phase. Now G2 phase, okay, it checks whether the DNA replication is correct or not. Okay, and DNA repair, whether it requires DNA repair. Okay, so that is checked by G2 phase. And also in G2 phase, certain proteins which are important for the next phase, that is M phase, okay, they are synthesized during G2 phase. Okay. So during G2 phase, what happens? The proteins which are required for the next phase, they are synthesized. And also it is checked whether DNA replication is completed or not, whether it is correct or not. If it is incorrect, then it is repaired in G2 phase. Now this is all about interphase. This is all about interphase. One more thing that you should remember here is during S phase, even the centrosome which contains the centrioles also divides in S phase. So DNA divides, you can say DNA is replicated, and also the centrosome also divides here. Okay. So once the G2 is over, then the cell enters the next phase, that is called M phase. That is mitotic phase. Again, this M phase is divided into two parts. One is called karyokinesis. Now this blue shading, okay, shade area, okay, it shows the karyokinesis. So karyokinesis means division of the nucleus. And the next part is cytokinesis. Karyokinesis, which is followed by cytokinesis. Now, during karyokinesis, or you can say nuclear division, there are four phases: prophase, metaphase, anaphase, and telophase. Now, in all these phases, what happens? The nucleus divides. That is, the chromosomes are formed and they are separated. Okay, in karyokinesis. So once karyokinesis is over, then the cell divides. Okay, the cytoplasm of the cell divides. So here only the nucleus is divided. Here, once the nucleus is divided, once the chromosomes they move to opposite poles in a cell, okay, then it is followed by cytokinesis. Cytokinesis is nothing but division of the cytoplasm, which ultimately results in the formation of two cells. So like this, from a single cell, two cells are formed. Okay. Now this is what we call as cell cycle. Now these cells which are formed, they enter G0 phase, and whenever required, okay, whenever they are required to divide, they pass to the next phase, that is G1 phase, and the process continues. Now these are different phases of cell cycle. There are basically two phases: interphase, M phase. M phase is divided into karyokinesis, cytokinesis. Okay, and karyokinesis is divided into again four phases: prophase, metaphase, anaphase, and telophase. Now, one thing that you should remember here is that even though the DNA, which is doubled now, okay, it was earlier 2C, now it has become 4C, okay, in S phase and also in G2 phase, okay, it will become 4C. But even though the DNA doubles, okay, but the number of chromosomes doesn't change. Suppose this is a cell cycle of a typical human cell, then there will be 46 chromosomes even though the DNA doubles. Even though the DNA doubles, the number of chromosomes do not change. Okay, it does not change. Now let's see how. Now, usually what you have seen, the structure of metaphase chromosome, you have seen that there are two chromatids here. There are two chromatids. So one I have labeled A, one more I have labeled A1 here. Okay, so there are two chromatids in a metaphase chromosome. But in reality, there is only one chromatid. In reality, there is only one chromatid, because you know very well that chromosomes, they are made up of DNA, right? They are made up of DNA. So initially, there is only 2C DNA. Okay, so DNA has not become doubled before S phase. Okay, where there is only one chromatid. Only one chromatid. Okay. Now, after S phase, so the DNA, okay, from 2C it becomes 4C. Then what happens? One more chromatid is synthesized here. You can see. That is why you see two chromatids in a metaphase chromosome. Okay, but actually there is only one chromatid. There is only one chromosome, which is made up of only one chromatid. But since DNA has doubled, even okay, the chromosome has doubled. Chromosome has doubled in the form of an extra chromatid. But both the chromatids form a single chromosome, and that is how, even though the DNA becomes double, the number of chromosomes will remain same. But the only difference is usually there is only one chromatid, but in metaphase chromosome there are two chromatids. Now these two chromatids, they are called sister chromatids because they are Xerox copies of each other. Okay, so they are exactly alike, but the only difference is one is the original DNA, the second one is duplicated DNA. Second chromatid contains duplicated DNA, so single chromatid. Okay, one chromatid contains actual DNA, okay, or you can say original DNA, and the other one contains the Xerox copy of the this original DNA. So A and A1, they are same. They are same, and they will be separated during the anaphase, so that each cell, so that each pole receives only one of the sister chromatids, either this or this. So usually, what I'm suggesting here is that usually there is only one chromatid in the chromosome before S phase. After S phase, another chromatid is synthesized because DNA becomes double. So chromatids also become double. Okay, right. Now let's see what are homologous chromosomes. Okay, now we have 23 pairs of homologous chromosomes. Okay, in case of man. Now homologous chromosomes, they are the chromosomes which are similar in length. Okay, they should be similar in length. Okay, and have the same position of centromere. Here you can see they have the same length, same position of the centromere, and also they should have same loci. They should have same loci. Here you can see. Okay. Now these are the loci. Loci are the places, or you can say the location on chromosome where alleles are present. Now here you can see, so there is an allele here. So this allele A. Okay. If on, okay, now this is one, these are a pair of homologous chromosomes. Okay. Now allele here, capital A, that is dominant allele, small a, that is recessive allele. Okay. Here you can see both are present on the same loci. Okay, allele A, capital A and small a, they are present at the same loci. At the same time, here you can see B and C also, they are present on the same loci. Then such chromosomes are called homologous chromosomes. So homologous chromosomes, they are the chromosomes which are similar, same in length, and have centromere at the same position, and also they should have the same allele loci. Then they are called as homologous chromosomes. In humans, there are 23 pairs of homologous chromosomes. In each pair, okay, in each, now this is a pair of homologous chromosomes. In each pair of homologous chromosomes, one is called maternal chromosome and other one is called paternal chromosome. So in a single set of homologous chromosomes, or you can say single pair of homologous chromosomes, one chromosome is called maternal chromosome, other chromosome is what we call as paternal chromosome. And here you can see the same homologous chromosomes. This is one pair of homologous chromosomes. Each homologous chromosomes, in case of in metaphase, okay, they have two sister chromatids. They have two sister chromatids which will get separated during the anaphase. So in short, what you have to remember is chromosome is usually made up of a single chromatid. Only after the S phase where DNA becomes double, an additional chromatid is produced. Okay, so the amount of DNA after S phase becomes double, but the number of chromosomes remains the same. But the only difference is each chromosome, instead of having one chromatid, it has two chromatids, and the sister chromatids, they are Xerox copies of each other. Now let's see mitosis. Now, mitosis is an equational division in which both the daughter cells will receive the same number of chromosomes. Now, this is the mother cell, and these two are the daughter cells. And it means the number of chromosomes in the mother cell, okay, it's same as the number of chromosomes in the daughter cells. Now, first phase is prophase. Now, during prophase, the nuclear membrane disappears, the cell organelles disappear, okay, and then the chromosomes, they start condensing. They start condensing. At the end of prophase, the chromosomes, they become condensed. Now, these chromosomes are formed from chromatin. These chromosomes are formed from chromatin, and you know very well that chromatin is made up of DNA plus histones and non-histone proteins. Okay, so during prophase, what happens? The cell organelles disappear, the nuclear membrane disappears, then centrioles, they start moving to opposite poles, and the chromosomes, the chromatin condenses to form chromosomes. So these are the events occurring during prophase. Now, during metaphase, that is next phase, the centrioles, okay, the centrioles, they are moved to opposite poles. Now, this is the location of the centrioles. So, centrioles move to opposite poles, and centrioles, they start producing, okay, they are already produced here, they already produced these microtubules. Here you can see. These are the microtubules which are produced by the centriole, which is present here. Okay. Now these microtubules, they form spindle apparatus. That is why they are also called as spindle fibers. Now during metaphase, what happens? The chromosomes, they get aligned themselves at the center. So only two chromosomes have been shown here just for representation. Okay. Now the chromosomes, they align at the center like this. They align at the center or equatorial line or equatorial plate, it is called. So there is nothing, okay, there is nothing called equatorial plate. It does not exist actually. So it is an equatorial line at the center. And at this point, okay, these microtubules or spindle fibers, they attach themselves to the centromere region, that is the kinetochore region of the chromosomes. So that is what happens during metaphase. So during metaphase, the chromosomes are completely condensed, and the centrioles have moved to opposite poles. Centrioles have produced spindle fibers which are made up of microtubules, and these spindle fibers are attached at the kinetochore region of the centromere. Okay. And the next phase, that is anaphase. During anaphase, what happens? Okay, the centromeres divide. The centromeres, they get separated. Okay. Now these spindle fibers, they pull. Okay, the spindle fibers pull the sister chromatids. So now this is a typical metaphase chromosome which has two chromatids, as you can see here. Okay. So in anaphase, what happens? These spindle fibers, which, okay, they separate, or you can say they pull the sister chromatids to opposite poles. Here you can see. So this sister chromatid comes to this pole. This sister chromatid comes to the opposite pole. The same thing happens with the other chromosomes also. So during anaphase, what happens? The sister chromatids are separated. They are pulled apart with the help of these microtubules or spindle fibers. And at the end of anaphase, these sister chromatids, they move to opposite poles. Okay. Now here you can see these are sister chromatids which have moved to opposite poles. That is what we call as telophase. Okay. In telophase, what happens? The sister chromatids, that they are moved to opposite poles. Okay. And their chromatin, okay, their chromosome start decondensing. That is, chromosomes, they start converting into the chromatin, and then again nuclear membrane reappears in the telophase. So at the end of telophase, you'll get two nuclei in the opposite poles of the cell. In the next phase, now this is all karyokinesis. Okay, prophase, metaphase, anaphase, and telophase. It all comprises. Okay, it's all karyokinesis. That is division of the nucleus. In the next step, that is cytokinesis. And in cytokinesis, what happens? Okay, now nucleus, the chromosomes have moved to opposite poles, and nuclei are formed at the both the poles. At the center of the cell, at the center of cell, okay, a furrow starts appearing. This furrow separates the two cells. Okay, so each cell having its own nucleus. There is nothing but cytokinesis. So the same cell, the cytoplasm of the same cell divides so that two daughter cells are formed. Okay, so from from the mother cell, two daughter cells are formed. Okay. What you have to note is the number of chromosomes is same in both the daughter cells. So these are daughter cells, this is the mother cell. So the number of chromosomes in mother cell is same as that of daughter cell. So here number of chromosomes is 2. Here you can see number of chromosomes is 2. Here also it is 2. The only difference is there were two chromatids here. Okay, in the daughter cell, sorry, in the parent cell, but we have only one chromatid. Okay. So instead of two, there is only one chromatid in the chromosome. Now, one thing that is not shown here is decondensation of the chromosome. So, in the telophase, at the end of telophase, these chromosomes they decondense to form chromatin. Now, this is mitosis in brief. Now, coming to meiosis. Now, meiosis compared to mitosis, it is a reductional division. So, in mitosis, the number of chromosomes in daughter cells is same as that of the parent cell. That is, the daughter cells receive the same number of chromosomes as that of parent cell. But in meiosis, which is a reductional division, here the daughter cells receive half the number of chromosomes. Half the number of chromosomes. Okay, compared to the mother cell. Now, just to example how mitosis is different from meiosis, just see this. Now, in mitosis, okay, if the amount of DNA is 2n, sorry, it is the number of chromosomes. So, diploid chromosomes, say for example, 46 chromosomes are there, then the daughter cells will have same number of chromosomes, 2n. Okay, its number is maintained here in mitosis. But in meiosis, here you can see a diploid daughter cell, okay, it will produce 4 haploid cells, which have half the number of chromosomes compared to the parent cell. Now, let's see how this happens. Now, meiosis is divided into two phases here, meiosis 1 and meiosis 2. Again, meiosis 1 is divided into prophase, metaphase, anaphase, telophase, etc. Okay. Now, let's see meiosis 1. So, the first phase of meiosis 1 is prophase 1. Now, in prophase 1, okay, these 5 important events occur: leptotin, zygotin, paketin, diplotin, and dikinesis. Now, these are the phases of subphases of prophase 1. Now, this does not occur in case of mitosis. Okay, prophase 1, okay, the events which are occurring in prophase 1 occur only in meiosis, not in mitosis. Now, these are the subphases of the prophase 1. Now, let's discuss these one by one. Now, first one is leptotin. Okay, so in leptotin, what happens? The chromatin starts condensing and they form thin thread-like chromosomes. They form thin and thread-like chromosomes. In the next phase, that is zygotin, the chromatin, okay, the chromosomes are condensed. In the next stage, that is zygotin, chromosomes are condensed, and the homologous chromosomes they form pairs. They form pairs like this. So the homologous chromosomes they start coming towards each other. Now this is, say for example, this is maternal chromosome and this is paternal chromosome. So this is a pair of homologous chromosomes. Like this, there will be 23 pairs of homologous chromosomes in case of humans. Okay. Now, during zygote, what happens? The chromosomes, they have become condensed, and these chromosomes, they align themselves. Okay, they align themselves together. They start coming together. Now, this process is what we call as synapsis. So, synapsis is a process in which the homologous chromosomes, they come closer and align themselves along the length of the chromosome. Now, this is what we call as synapsis. Okay. Now, during synapsis, so each pair of so this one pair they are shown here. Okay. Each pair of homologous chromosomes, now it is called, okay, during zygote, or you can say during synapsis. So once the two chromosomes or the two homologous chromosomes, they align themselves, this is what we call as synapsis. Okay. The synapsis has two chromosomes. Synapsis has two chromosomes, a pair of homologous chromosomes. That is why these are called bivalents. These are called bivalents. Then there are two chromosomes in a pair. They're called bivalents. So like this, there are 23 bivalents in case of humans. There are 23 bivalents. Okay. Now, they are also called tetrad. They are also called tetrad because here you can see 1, 2, 3, 4. There are 4 chromatids in each pair of homologous chromosomes. That is why they are also called as tetrad. They are called bivalents because there are 2 chromosomes in a homologous pair. They are called tetrads because there are 4 chromatids, 2 chromatids each for each chromosome. Now, let's see what happens during synapsis. Now, during synapsis, between the homologous chromosomes, here you can see these are paternal chromosome, this is paternal chromosome, and this is maternal chromosome. So both have they have been shown in different colors here, just for sake of understanding. So when the homologous chromosomes, they come together, they come closer. Okay, between these chromosomes, a complex called synaptonemal complex is formed. Okay, during synapsis, when the two homologous chromosomes, they come closer. Okay, between the chromosomes, a protein complex is formed. A protein complex is formed. This protein complex is what we call as synaptonemal complex. This synaptonemal complex keeps the homologous pair of chromosomes together. Here you can see this is the paternal chromosome, this is the maternal chromosome, and these two are sister chromatids of the maternal chromosome, and these two are sister chromatids of paternal chromosomes. Then these chromosomes, they are held together. Okay, during synapsis, these chromosomes are held together by, here you can see, the protein complex, a complex which is made up of different proteins. Now, this complex is what we call as synaptonemal complex. Okay. So, synaptonemal complex holds the chromosomes together during zygote. Okay. So, in leptotin, the prophase 1, okay, the first phase is leptotin. During leptotin, the chromatin is converted into, or you can say it starts condensing into thread-like chromosomes, and it forms thread-like chromosomes. In the zygote, the chromosomes are condensed. Homologous chromosomes, they start pairing, they start coming towards each other to form synapses, and during synapses, a synaptonemal complex is formed between the homologous pair of chromosomes. And since there are two chromosomes in each homologous pair, okay, now it is called bivalent, and since there are four chromatids, it is also called as tetrad. The next phase is pachytene. Okay. Now in pachytene, the non-sister chromatids, okay, during in pachytene, the non-sister chromatids, they undergo crossing over, they cross over each other. Now let's see this process in detail here. Okay. Now let us see the same pair of homologous chromosomes here. Here you can see there is overlapping. Okay, so this is, say for example, this is maternal and this pink one is paternal. Here you can see that this paternal chromosome, okay, it has uh, it has crossed, okay, or you can say it has overlapped the maternal chromosome at this point. Okay. Now, let's say these two are the homologous chromosomes. Okay. Now, the non-sister chromatids overlap. Remember, not these two. Now, these two are sister chromatids of the same homologous chromosome. These two are sister chromatids of the same chromosome. Okay. But, so this is blue. Okay. Let's say this blue is maternal chromosome, this red is paternal chromosome. Then, this one blue and one red, okay, chromatids, now they are non-sister chromatids. Remember, only the non-sister chromatids, they cross over each other, they overlap each other, not sister chromatids. You don't see any overlapping between blue and blue. You don't see any overlapping between red and red. So the overlapping is between the blue and the red chromatids. So blue chromatid represents maternal chromosome, red represents paternal chromosome, or vice versa. So during packaging, crossing over occurs. Now, during crossing over, the non-sister chromatids, they overlap each other at certain points. Okay, now at certain points, and the point where the crossing over is taking place, it is called as chiasma. Okay, chiasmata, singular chiasmata. Sorry, singular chiasma. Chiasmata is plural. So, what is chiasma? Chiasma is a point where the non-sister chromatids of the homologous chromosomes, they overlap. Okay. Now here what happens? Crossing over occurs. Now during crossing over, okay, few genes from one chromosome of the homologous pair, they come to the other pair, or you can say the genes which are located on maternal and paternal chromosomes, they are exchanged. Let's say this is the maternal chromosome and this is the paternal chromosome. Now here what happens after crossing over? What you can see here is that, okay, a part of the paternal chromosome has moved to maternal chromosome, and a part of maternal chromosome has moved to paternal chromosome. So some of the chromosome segments containing alleles, they are exchanged between the two chromosomes, so that some genes from the maternal chromosome move to paternal chromosomes. Some genes from the paternal chromosome move to maternal chromosomes. Now, this process of crossing over thus results in recombination. Okay. Now, this exchange of genes between the non-sister chromatids of the homologous chromosomes, okay, is nothing but recombination. So, recombination is the exchange of genes, and crossing over is a phenomena that results in the recombination. Hope you understood this. Okay, now let me explain once again. Packeting. So during packeting, okay, the paired chromosomes, they cross over each other, overlap each other. The non-sister chromatids of the homologous chromosomes, they cross over each other. Okay, and during crossing over, parts of the chromosomes containing genes are exchanged between maternal and paternal chromosomes. That is called as recombination. And the point where the crossing over is occurring is called chiasma. It's called chiasma. Now, in the next stage, that is diplotin, okay, the recombination is complete. So recombination starts in packetin. At the end of packetin, the recombination will be complete. Then in the next stage, that is diplotin, the homologous chromosomes, they start moving, okay, they start moving away from each other. Okay, so after recombination, the homologous chromosomes, they start moving away from each other. Okay. Now in the next phase, that is dikinesis, the homologous chromosomes have moved away, but still they are closer at certain points. Okay, so in dikinesis, okay, the homologous chromosomes are almost separated, but still they will remain together. But in diplotin, what happens? They start moving away from each other, but they have completely moved away here. Still, they remain together in diakinesis. Now, the next phase is metaphase 1. Now, during metaphase 1, here you can see these are the recombinant chromosomes which have exchanged their genes. Okay. Now, you know very well that during diakinesis, even though they are separated, even though they are moved away from each other, but they are still together at certain points. Okay. Now, these are bivalents. Okay. Now, during metaphase 1, the bivalents, that is each pair of homologous chromosomes, they are arranged at the equatorial region. Okay. So, the tetrads, they are given here. The tetrads or bivalents, they remain at the center of the cell in metaphase 1, and here these are centrioles. They have moved to opposite poles, and centrioles have produced these fibers, spindle fibers or microtubules. These spindle fibers, okay, they are attached to the chromosomes, you can say the bivalents. Now this is different from metaphase of mitosis, because in metaphase of mitosis all the chromosomes they align at the center, not the bivalents, because there is no synapses occurring, okay, in metaphase of mitosis. But in meiosis, what happens? Chromosomes, they remain in pairs like this. So like this, there are 23 pairs in case of humans. They are aligned at the center. Okay, right. This is what happens in the metaphase. So the main difference is there only individual chromosomes are aligned at the center, but here bivalents are aligned at the center. Next, in anaphase. In anaphase 1, remember, in anaphase 1, only homologous chromosomes separate. Now, this is one part of homologous chromosome, this is another partner of homologous chromosome. Only the homologous chromosomes move to opposite poles. So, homologous chromosomes move to opposite poles. Okay, so these are the homologous chromosomes. Only they are separated, but sister chromatids they remain together. You can see. So these sister chromatids are not separated, but usually in metaphase of mitosis, okay, in metaphase for first stage only, there is no second metaphase, of course. In mitosis, what happens? Sister chromatids separate, but in metaphase 1 of meiosis 1, sister chromatids do not separate. Only the homologous chromosomes separate. Only homologous chromosomes separate. Okay, here you can see. So these spindle fibers to start pulling the homologous chromosomes to the opposite poles, so that these partner chromosome goes to one pole, the other partner goes to other pole. Okay. Now what you should observe here that in telophase, okay, not at the after anaphase, telophase follows anaphase 1. Now in anaphase 1, what happens at the end of anaphase 1 or the telophase? This pole has 23 chromosomes. This pole has 23 chromosomes only. Okay, but in anaphase of by the end of anaphase of metaphase, okay, each pole had 46 chromosomes, but here each pole has only 23 chromosomes, which is half of the parental cell, half of the chromosome of the parental cell. Okay. The next phase is telophase 1. Okay, in telophase 1, the chromosomes which have moved to opposite poles, they decondense, they decondense, okay, to form nucleus. Here you can see the nuclear membrane is reappearing, and this chromosome is converted into chromatin and centrioles. Okay, they are found here. Okay, they start degenerating, means they start degenerating their microtubules. Okay. Now this is telophase 1. Okay. Now you can see in telophase 1 there are two cells here. There are two cells. Next comes prophase 2, that is meiosis 2. So far we discussed about meiosis 1. Now next comes meiosis 2. Meiosis 2 is divided into again several phases: prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. Now we already know that there are two cells which are formed at the end of telophase 1. Now these are the two cells. Okay. Now let us consider different phase of the each cell. Now same. Okay, each cell follows the same pattern here. Each cell, okay, it passes through metaphase 2, anaphase 2, telophase 2, and cytokinesis phases. Now you know very well that at the end of prophase 1, okay, at the end of anaphase, okay, what happens? Each pole, okay, so this was one pole, this is another pole. Okay, each pole has only, okay, homologous chromosomes. Okay, homologous chromosomes separated. Now this pole has one set of homologous chromosomes. This has another set of homologous chromosomes. In the metaphase, these homologous chromosomes, okay, not homologous chromosomes, okay, we cannot say homologous chromosomes now because they are already separated. Now these chromosomes from each cell, they align at the equatorial line, and spindle fibers, they attach, okay, to the chromosomes. Same thing happens here also. In anaphase 2, the sister chromatids separate. Okay, in anaphase 2, sister chromatids separate. Here you can see these sister chromatids are separated in anaphase 2 of both cells. Now this is the difference. One more important difference between mitosis and meiosis. That is, in mitosis, in a single anaphase, the sister chromatids are separated. But in meiosis, okay, in the first anaphase, anaphase 1, only homologous chromosomes separate. But here in anaphase 2, sister chromatids are separated. So in anaphase 1, only homologous chromosomes separated, and in anaphase 2, the sister chromatids of the chromosomes are separated. So each cell, okay, so each cell which is formed, okay, after meiosis 1, it enters the meiosis 2, and each cell, it forms two cells. Now these chromosomes which are separated, they start move to opposite poles, and then they start decondensing, and cytokinesis splits the two cells here and two cells here. So this cell produces two cells, this cell also produces two cells. So at the end of meiosis, four cells are formed from the daughter cell. Four cells are formed from the daughter cells. So in meiosis 1, two cells are formed. In meiosis 2, these two cells give rise to two more cells. Now all the four cells, they are haploid because they have half the number of chromosomes compared to their parents. Now in the next clip we shall discuss some questions.